All right, ladies and gentlemen, this is going to be the next video on proteins. So let's get started on this next video. So what is a protein? Well, you might think of a protein as something like fish or nuts or eggs or meat of some sort. And really, proteins are going to make up the structure of our body. They're playing a crucial role in making up the structure of our body. And these proteins, if we were to break them down, we would be able to see little tiny amino acids. And these amino acids are the main backbone in the structure of what we go through and talk about as a protein. Now, they're going to serve a couple of different purposes in our body. They're going to really make up the main parts of cells and the solid material. They're going to help cells divide, and they're also going to help the immune system. But when we're thinking about proteins, it's really that like solid structure or any of the carbon structure really in our body. So proteins, when we go through and talk about them, are made from amino acids. And an amino acid is a simple organic compound containing a carboxyl and some sort of amine group, which are really the building blocks of the protein chains. So when I say amino acid, I'm talking about a monomer. Now, if we were to look at amino acid structure here, what we can see is we're going to have our amino group, we're going to have our carboxyl group, and then we're going to have our R group here, which is our side chain, which allows us to go through and it's going to essentially be what our amino acid is attached to, whatever other structure. So if we're looking at this, each amino acid has a central carbon atom, and then there are 4 other atoms bonded to it, which allow for the functional groups, the amino group, the side chain, the carboxyl group. And this is going to be what we consider the functional groups of this specific amino acid. So when we discuss proteins, we have amino acids, and these are going to create chains of amino acids, and we call these polypeptides. So when we're talking about this, polypeptides are chains of amino acid held together via peptide bonds. Now, as we continue to go, we can take those peptide bonds, and these chains are going to fold into complex 3D structures that we know as proteins in animals. Now, we can then go through and eat other animals or eat other proteins from plants, break them down in our stomach, bring them back to our polypeptide chains, and then rearrange them into the human body. So one, our body's going through and taking these peptide bonds and arranging them into complex 3D structures that make up the human body. And then 2, our stomach's breaking down these complex structures from other animals and plants to create these peptides to go through and allow for rearrangement in our body. So if we were to look at this from a hierarchical standpoint, we would have amino acids being our monomers, peptides being our polymers, and then we can fold those peptide bonds and allow for those peptide chains to be complexly folded into what we know as a protein. So when we talk about amino acids, there are 20 amino acids total, and this can essentially allow for infinite proteins via the arrangement of these amino acids. And when we're discussing types of amino acids, there's 2 main types. We have essential amino acids, and we have non-essential amino acids. So when we're discussing essential amino acids, these are amino acids that can't be made in our body. There are 9 that the human body cannot synthesize, therefore we have to get them through our diet. Now, if we're looking at this, there are also non-essential amino acids, and these can be made by our body. There are 11 of these amino acids that the body can synthesize and make. So as we look at our 20 amino acids here, we have the non-essential amino acids, which I will go through and highlight in green. Examples of this are going to be things like tyrosine, aspartic acid, cytosine. And we also have our essential amino acids. And our essential amino acids are going to be here highlighted in pink. And remember, there are 9 essential and 11 non-essential. So you have histidine, proline's going to be 1. All of these in the pink are going to be our essential amino acids that we cannot go through and synthesize ourselves and are necessary for us to consume in our diet. Now, there's going to be some that are what we call conditional. And conditional depends on what we're going through and what we're consuming in our body. And really it depends on the environment and depends on what diet we have and what specific animal or mammal we're going through and discussing. So why does all of this matter? Well, you need certain proteins from either animals or plants. So again, we're digesting these proteins to go through. There are enzymes that will go through and break up these polypeptide chains, and then we're going to rearrange them into what we consider our body. So we can eat an animal-based diet and a plant-based diet and get the same amount of protein that's necessary for us to go through and live. So let's say we had an animal-based diet where we had 1 serving of Greek yogurt, salad and chicken, and then 3 to 4 ounces of fish. We would get 64 grams of protein. Now, if we were eating a plant-based diet, well, we could have 1 cup of scrambled tofu and a slice of peanut butter and jelly toast. We could also eat 1 cup of shelled edamame, and then at dinner we could eat 1 cup of lentils and 1 cup of brown rice. Again, we are still getting the same 64 grams of protein, we're just getting it through 2 different diets. So we can also go through and look at polar versus non-polar amino acids. And I'm not going to spend time going through and explaining because we don't necessarily have to go through and memorize this. You can look this up in a textbook. But we can see the non-polar R groups in the blue, the electrically charged amino acids in the yellow, and then we have a polar R groups here in the orange. So polarity is going to be really, really important for the function of how the enzyme works. So let's say we had water here. Now water is going to affect the shape of the protein, and remember our enzymes are going to change shape based on our polarity. So let's pretend we had an enzyme here. The red amino acids are nonpolar, the yellow are polar, and the pink are neutral. Well, they're all going to interact with each other, and the way that they interact is going to change their shape and their folding. So polar and nonpolar R groups interact with the environment, which affects the folding. So how the proteins are folded is going to change the shape and change the function of those enzymes, which again are just proteins that allow for specific function in our body. And also, different cell parts have different shapes, and dependent upon how these polar and nonpolar amino acids interact, it's going to affect the shape of the protein. It's also going to be really important when we talk about polarity for how enzymes function and break apart substrates. So if we're looking at the 4 layers of this 3-dimensional structure that is created via polarity, well, we're first going to have primary structure. And this is just the sequence of peptide bonds that go through and form when we're talking about polypeptides. Now the second is going to be the secondary structure. And we can go through and arrange the backbone of these polypeptides into an alpha helix or beta-pleated sheets. Now, thirdly, we can get to tertiary structure. And tertiary structure is the combination of the alpha and beta sheets and helices to create the 3-dimensional protein structure that we see. Lastly, we can get to our quaternary structure, and this is the overall structure of the protein that we're going through and discussing. So if we're looking at primary structure here, again, we're having peptide bond formation via dehydration synthesis. And all a peptide bond is a covalent bond formed by joining the 2 carboxyl groups via the dehydration synthesis. So as we go through dehydration synthesis, we are going through removing a water and allowing these 2 monomers, or these 2 amino acids, to bond via peptide bond, create a polypeptide. So 2 amino acids are now linked in a chain, and we can continue to link them together in chains to create our polypeptides. Now the secondary structure, this is referring to the local folding of the backbone. We have our alpha helix, and then we also have our beta-pleated sheets. So if we look at this, the helices kind of look like this in the picture, and in an alpha helix sequence, the amino acids are hydrogen bonded, and these are going to create this helice that we see with our chains. Now, beta-pleated sheets are going to look like this, and they are more segmented polypeptide chains. And again, this has to do with hydrogen bonding and how the polar and nonpolar regions of the amino acids interact. When we go through and talk about this, these 2 backbones are going to make up our tertiary structure, and they're also going to make up our quaternary structure. Both are held together via hydrogen bonds, and the arrangement of these hydrogen bonds go through and create the helices or the sheets. And when we talk about tertiary structure is really when we start talking about 3-dimensional structure of that protein. And there's three things that are really going to go through and affect this. This is where the hydrogen bonds, the polar and nonpolar interactions with the environment and with other amino acids, and then also disulfide bridges are going to create this three-dimensional structure. So again, this is why polarity is important when we're talking about these side chains in these amino acids. Now, quaternary structure, this is going to be, again, an overall three-dimensional structure, but now we're talking about the whole protein. And this is also going to be affected by polar and non-polar regions and their relation to the environment. So we can see subunits here that now are bonded together, and this entire complex is what we call a protein. So when we're talking about quaternary structure, we're really describing the complete structure of that protein. So let's go back to denaturing. Remember, enzymes are proteins. Proteins go through and combine, and they make up our body, and they have important functions in our body. So why are extreme temperatures and acids dangerous? Well, if our entire body and all the solid carbon structure is really made out of these proteins, and specifically when we talk about enzymes, remember, denature is destroying basically those proteins. Those proteins are disintegrating and falling apart. That's going to have really, really negative effects on your body. And high temperatures and acids cause the proteins to denature, or extreme alkaline or basic environments are going to cause the proteins to denature. So if we go through, what this is doing is it's denaturing the protein. We are separating all the bonds between our amino acids, and temperature is what's going to cause that to happen. So this is why it's super important to be on top of a fever, because you want to make sure that you don't have a fever of over 104 degrees Fahrenheit, because it can cause enzymes and other proteins in your body to denature. So did you guys learn? Well, did you learn a couple of things? Did you learn what a protein is? Did you guys learn the protein structure? Did you guys learn about what an amino acid is? And then did you learn about how all of these different structures and functions relate to the human body? This is going to be the end of the video. I will see you guys in class.